Hello and welcome to my sixth video on IB psychology. Today we're talking about the biological perspective. My name is Linnea Karlberg and I'm an IB 12 student. Today we're talking about pheromones. If you pause the video right now, you can read what the IB requires you to know about this. Pheromones are chemical signals evolved for communication with other members of the same species. You could refer to it as transferred excitement. Pheromones were identified in 1959 by researchers Carlson and Lüscher in Germany, and this was called bombicol, which was found in butterflies. Pheromones are secreted outside the body, and they're believed to be released by the apocrine glands found in the armpits. Pheromones can be found in all animals, and it's usually detected through smell. There are two different effects of pheromones. First, there's a releaser effect, and this is a particular stereotype reaction, such as recognizing a species is ready to mate. But some also result in a primer effect, meaning a developmental effect, like putting a banana next to an avocado and helping it develop. And these reactions can happen anywhere, even when inappropriate, and some pheromones produce both effects. And the responses are innate, so not influenced by learning. And a study that looked into this is Wedekind and Fury et al. In 1997. They looked into the relationship between similarity of major histocompatibility complex and attractiveness of scent. So these MHC genes are responsible for immunological functioning. So this was a T-search smelling study in which T-shirts with the most appealing odors would belong to the participants with the greatest variation of MHC relative to the individual. And genetic differences in MHC genes are associated with advantageous immune functioning. So this study involved 120 male and female participants that assessed the odors of 6 T-shirts worn by 2 women and 4 men for 2 days straight. And it was found that the preferred scent was worn by people with the most different MHCs. So individual odor plays a role in mate choice and is gender specific. And individual odors may differ as a result of both differences in MHC and from differences in pheromones. So this study supports the role of pheromones in affecting behavior. However, it takes quite a reductionist view of the complex concept of human behavior of mate choice. And the reasoners are not quite sure if the Confederate actually wore the T-shirts and followed the instructions, which included don't wear deodorant, don't wear soap, anything that smells. I'm sorry, don't use soap, of course. And Dr. Wyatt, he has actually come out and said, these studies are about individual odors and the possible genetic compatibility, sure, but they're not about pheromones molecules identical in all males and in all females. Quite long ago, in 1976, a researcher named Russell did a lot of research and experiments into pheromones. One of them being about mother-baby interaction, which is believed to be guided by pheromones, and it just shows that human response to pheromones begin early. So this study sampled 10 breastfeeding mothers and their babies, and the babies were to smell breast pads, their mothers, a clean one from a stranger, and one in cow that was induced into cow milk. Russell then measured the intensity of sucking when exposed to the smell of the pads, and it was found that babies could not tell their mother's pad until they were 6 weeks. After that, 6 out of 10 babies got it correct. And this was then believed to show that humans have pheromones, and humans are affected by them from a young age, and that it affects mother-baby interaction because the babies recognize the mother's pheromones. However, the sample here was quite small, which makes us question the generalizability, and there's surely some triangulation necessary, since this was performed very long ago. Another study that was also a T-search smelling study, they're quite iconic for this area, was that when Russell was trying to show that humans are affected by pheromones to 16 males and 13 female college students. And once again, they wore a T-shirt for 24 hours this time, and they were asked not to wash or use deodorant during this time. Following, the participants smelled the T-shirts, one of their own, an unknown male, and an unknown female. And the task was then to identify your own shirt and which one was female and which one was male. 13 males and 9 females correctly identified the shirts, which made Russell suggest that humans have a system that identifies pheromones which affect attraction. Well then, how does this really measure attraction? It seems like very heterosexual bias here in this study. And also, there are plenty of confound variables involved, like male sweat has a stronger smell than female sweat. And also, female shirts were rated as more pleasant, which once again shows that it probably has more to do with the smell of the sweat. Is this really pheromones communicating through species? We're not quite sure. So Doty et al. Actually did an attempt to show that social conditioning might be responsible for the results that was found by Russell. So male and female sweat was collected on gauze pads, and they were then given to male and female participants in sniff bottles. And the instructions here were that some of the bottles were male, some female, and others a mixed. But in reality, there was one study where the sweat was only from males, another was only female, and some was no sweat. And there was also a condition or a study where they were mixed and female only, and some with no sweat. This was, of course, then to show that it's probably that male sweat just smells more. So yeah, Doty concluded that when smells were from both males and females, about half the participants guessed correctly, and participants rated the stronger smells as male and the weaker and neutral as female. And no differences was found between male and female smell ratings when smells were equally incubated. So smell identification could easily have occurred from social conditioning through which gender smells the most, to put it simple. Well, evaluating this pheromone theory overall, first of all, there's this vomeronasal organ, VNO, which is contained in the receptor sites in animals. We don't know if the VNO of humans is functional, but this is the body part that in animals is responsible for pheromones and detecting them. The areola is also a possible discovery of human pheromone which should be looked into more. If secretions from a lactating mother's areola are placed under a baby's nose, the baby will initiate nipple-seeking behavior and sucking. Dr. Wyatt says that identifying and synthesizing a breastfeeding molecule could have a major significance, as some babies and mothers fail to execute proper breastfeeding, and the application of a pheromone could facilitate the process. However, to be certain about the existence of pheromones, we must identify a specific behavior or physiological response that is mediated by a specific chemical in the species. We also must isolate and examine the makeup of the molecules of the chemical, and must confirm that the molecule under the study is enough at natural concentrations to produce a releaser or a primer effect. And the studies examining this problem have not followed these steps, so they are simply inconclusive. We don't know yet if this is existing. Thank you so much for watching. Let me know if you have any questions.